Hello, welcome to the de.NBI ELIXIR Germany and GHGA knowledge series. I can present the organizers for those of you that they are not familiarized with us. So we are de.NBI ELIXIR Germany and GHGA. I start with de.NBI. de.NBI is the German National Bioinformatics infrastructure, the German network for bioinformatic infrastructures, and this is the national node for the European initiative ELIXIR, ELIXIR Europe. And as you can see in the map on the slide, it is a large network with a lot of with the main academic research infrastructure centers in Germany, across Germany, and they they deliver a lot of services, and they cover a lot of diverse areas related to bioinformatics, so like human, plant, microbial, proteomics, and other data services. One of the key services of de.NBI is the de.NBI Cloud. So they deliver compute resources for academia in Germany, and they also develop and deliver diverse tools and services such training for the life science. And you can visit the website if you wish more information about de.NBI. And now I have the pleasure to introduce GHGA. GHGA is the German Human Genome-Phenome Archive, and we are the German secure omics data infrastructure. We provide fair and controlled access to data, and in doing so, we ensure data security and make the data available for research. We are an NFDI consortium coordinated at the DKFZ in Heidelberg. We are a federated network of 6 data hubs across Germany. And we are also international connected. We are the German node to the European initiative, like the federated European Genome-Phenome archive and genome data infrastructures. And yeah, if you wish to know more about us, you can also visit our webpage. Now, our speaker today, Jisung. Jisung started as a researcher support specialist and then promoted to a senior software engineer. Previously, he worked at the term in Australia. His job focused on specialized software solutions and data platforms to support large-scale ecological and environmental research. And nowadays, he's a research software engineer at EMBL in Heidelberg, and he's involved in European open science initiatives like EOS Clive or NFDI for Bioimage. And his work nowadays focuses on cloudifying workflows and containerizing bioimage analysis software. And this is the talk, and this is the topic of today. So thank you very much, Jisung. The floor is yours. It is a pleasure having you with us.
So hi everyone. So thank you for joining this talk. So I'm E. I'm a research software engineer working in the bioimage analysis support team at EMBL. So today I'm going to talk about how we do cloud-based bioimage analysis at EMBL. So it's not about a specific software solution, but it's more general introduction of how we do image analysis at EMBL. So first of all, I would like to start with this roadmap. So as you can see from left to right, so this is the bioinformatics roadmap from image acquisition from microscope and to the downstream analysis using various type of software and platforms. So basically, we acquire image from the microscope. And we try to convert different types of images into a OMIZAR format and put the original one on the tape and then in archive. And the images we aim to store all the converted image in a S3 cloud compatible storage. And then we also have this lab information management system, and we publish the metadata into Bioimage archive. So based on the images in the bucket, and we perform various type of analysis. So which is the center part, and all the analysis is backed by different technologies such as Kubernetes, OpenStack, and containers. So we also try to publish our workflow to Workflow Hub. So today I'll be focused mainly in the center part about the analysis. So to start with, I would like to show some typical images that we work with here at EMBL. So first type of image is fluorescent images. So as you can see, this is a hair cell image, and the green one is a nuclear envelope, and the red one a DNA. So this is a time-lapse image. So we take this every 8.5 minutes and keep doing that for 44 hours. So in the end, we have 329 time points. And this is a very high dimensional data set. So you can find the detail, everything from this RDR link here. And the next type of image is electron microscopy images. So this is another example from my colleague Julian. So this is also a HELA data set. So this is relatively small. So the whole volume is about 90 plus gigabytes. And then for this data set, we have 1,727 slices. So the images are aligned using a software called AMST, also developed by Julian. Okay, so we mainly work with these two different types of images, but what are the characteristics of these images? So they both have very large size. So sometimes you can easily above 100 or 200 terabytes. And for EM images, and the resolution is extreme, so in nanometer. And fluorescent images are very high throughput. So usually it's very high dimension, up to 5 dimensions. So EM is usually 3D volumes. So both images come with metadata. So for example, the settings, pixels, etc. But this is not always by default, and it also depends on the person who acquired the images, whether they are putting metadata or not. So, and another one is that these images are usually taken by biologists, so who really want to focus on the science, not maintaining or managing this large science data on the compute cluster or laptop. So given all of this characteristics of different types of images, so we're facing some challenges. So the main challenges, first of all, so is about scales, as I mentioned. So both types of images are very large. And then the formats. So there are currently more than 200 proprietary microscopy image formats out there. So it's a maintenance nightmare. So you see from the right, there's an example showing different types of microscopy images. And another challenge is the access. So quite often that only a subset or subregion of this entire volume is of interest to the researchers. But to do that, so you have to load the entire image into memory, so which is very challenging or very difficult to do it with your laptop. And also there's no parallel access. So if like a lot of workers trying to write or read the same file, that creates an I/O bottleneck. So how do we solve this challenges? So, and the OMIZAR is the solution. So that's why we at the beginning mentioned we try to convert all the images into the standardized format OMIZAR. So you can find a lot of information on the web. So I won't go into too much detail about that. So basically, OME is the OME specific. ZAR is the image format. OME is the metadata, and it solves the challenges we mentioned in the previous slide. And then everything is chunked in OMIZAR. So you can, for example, if you're only interested in this specific region, then you can just stream this region from cloud storage. You don't have to load everything into memory. And then these are the SAR array data, and then plus the metadata here. Okay, so we're trying to convert everything to WebSAR, but there are a lot of tools can do that. So this is just a very brief introduction about the tools that we use to convert to WebSAR format. So UV-Bridge and Batch Convert are developed by boosted by Bioimage, and UV-Bridge is a newer tool. So it supports conversion from TIFF, for example, to WebSAR images. You can run locally and also can run on your institutional cluster, like Slurm cluster. And another one, the next one is NGFF converter. This is a desktop GUI application from Glencoe software. It gives you an interface. So if you don't want to create your own commands to talk to the cluster, you can use this. And then basically it wraps the library called Bioinformatics to RAW. So you internally use this library to do the conversion. Okay, so now we try to put OMSR in the cloud compatible storage, for example, S3, and then based on the OMSR image, we're trying to do the analysis. So that's the analysis part. So in our opinion, so cloud-based image analysis basically has these three different types of tasks. So from left to right, so the interactive analysis, which means you typically require a GUI software, and then some examples like virtual desktop or some browser tools or Galaxy interactive tools. So you interact with the GUI application. And the middle one is interactive scripting. What we mean by that, so this is useful when you want to, for example, visualize your data, or you want to test something before you actually submit your workflow. And the last one is batch analysis. So this is like you automate everything. So you build your workflow into different steps and you chain them together, pre-configure everything, and then you put your parameter and then start. Everything is automated. And some example tools like Galaxy and Nextflows. So for today, I'll be focused a little bit on virtual desktop for interactive analysis, and then Nextflow and Galaxy on the batch analysis. Okay, let's start with the batch analysis first. So, so as we just mentioned, so batch analysis means we automate everything. So, and then at EMBL, we mainly use Nextflow for this purpose. So, because first of all, it's portable, so it can be run very easily on a local laptop or on cluster, or on the cloud with very minimal configuration changes. And they're reproducible. So every process in a Nextflow workflow, the dependency of each process are containerized. So that makes sure the environment the software runs is fully reproducible. So, and the last one is parallelism. So it handles the parallel processing internally automatically. So you don't have to write code or specifically write code for this. And the other workflow management system that can do this batch analysis is Galaxy Anonymous NameMake. We mainly use Nextflow with a little bit of Galaxy. So this is an example Nextflow workflow, the Institute Sequencing workflow created by Nima, so my colleague. It's just an example. So the input is OMSR, and then the registration, some pre-processing, for example, tiling, formatting, and then we use the Starfish decoding for the results, and the Starfish is containerized. Another example that we use the Galaxy is this one. So this one trains a neural model for image segmentation. So similarly, the input, so in Galaxy, it's called data collection, but it can be a list of TIFF or different format or OMSRs. And then pre-processing and then convert to the format that any labeling recognize. So any labeling is a interactive annotation tools. So the difference, the distinction of this workflow is that any labeling is interactive. So in the batch analysis, the user have the options to interact with the annotated images. So it's kind of human in the loop workflow. And you can find more details in the workflow hub about this. Okay, so Now, what batch analysis can do? So first of all, there's no visual quality controls. So you don't know the failure until the workflow finishes and you look at the results. And there's no interactive parameter settings because you have to set everything before the workflow runs. So, for example, you never know the segmented threshold before you actually run this workflow on one of the images. And the last one, so there's no very limited interactive image annotations. So we mentioned in the Galaxy workflow that you can do limited annotation, but that's still not like a fully interactive. And then so for all of this, and then what we need next is an interactive analysis. So that's why we're going to start talking about this interactive analysis. So for today's talk, I'll be focused a little bit on the virtual desktops. So it's one of the tools that can perform interactive analysis. So a little bit of motivation why we create this virtual desktop, because we want to replicate the user laptop experience. And we want to use remote desktop technology, for example, VNC. And so from the user point of view, you get a desktop in your browser, and there's nothing else you need to do. Everything, all the software is pre-configured, pre-installed. So you can just open a browser, connect to the internet, and start using this. There are many different types of virtual desktops. So at EMBL, we mainly use two. So one is called BARD, one is called BAND. And the other virtual desktop for the NeuroDesk, Bioimage from the EPFL, and then the Galaxy Interactive 2. So the difference between our virtual desktop, the BARD and BAND, and the rest, except the NeuroDesk. So Bioimage and the Galaxy IT, the desktop environment is also in the browser, but they have one gigantic container with the desktop environment together with all the softwares. But what we do differently is that the application and the desktop environment is separate, so it's very easy to maintain. Now let's look at the first desktop environment called BAND. So BAND is originally based on the work done in Australia, so specifically the massive facility in Monash. And it was originally a Bioimaging contribution to the EOS Live project. The BAND entire desktop is based on OpenStack, and you deploy the use and configure it using Ansible. So currently we have, as far as we know, we have 3 different instances. We maintain the first 2. So we have a Tübingen instance which supported by de.NBI Tübingen node. This one is for everyday use. So, and the next instance is supported by CESNET in the Czech Republic, and also supported by Bioimaging. So this instance is only for course, so only available when there's a course happening. And the third one is from the Flemish Bioimage core in Belgium. So we don't maintain this, but they have their own instance of the BAND. So this is a technical architecture about the BAND platform. So as you can see, everything is based on OpenStack. And then basically it has 3 different types of layers. The web layer, so it has the web server, Tomcat, NGINX, and have this guacamole, which convert your VNC session into HTML5 and then make it available in the browser. And then for authentication, we use Keycloak, and we also maintain our virtual LDAP server to store some user information. And then there's a customized script to create a user account once user logged in, so everything is automated. And the core of the BAND is this virtual Slurm cluster. So these numbers are specific to instance. So, but yeah, we have GPU nodes, and then some instances we have both GPU and CPU nodes. And we also have this NFS storage mounted to the cluster. So BAND also provide access to the outside world. For example, you can mount your own cloud into the BAND desktop, or you can mount any S3 bucket. Okay, this is what the BAND desktop will look like. So you first come to the login page. After you log in, go through the login process, you come to this page. So in the middle of that, you can configure what do you need. So you can configure how many CPUs, RAM, or whether you need GPU or not. So the data within the BAND desktop is persistent. So for example, you start with 2 CPU, 16 gig, but later you found out it's not enough. You can just come back and then stop and then launch a bigger one with more RAMs. Your data is safe. So once you go into your desktop, this is what you see in the browser. So this is the band desktop. So you found all the applications like data access, image analysis, and everything here in the menu. This is a cell-pose example, cell-pose application doing some image segmentations using GPU. And all these applications are containerized using Singularity or Optinite. So for data accessing band, we provide a lot of different clients. So remotely, we have Omero client, XNet, and S3. But locally, it's also possible to use local storage for analysis, but the space is very limited. So mainly locally, you have scratch space and user hole. So if you don't want to use remote, and then you have to stage your data in band. Okay, brief introduction about band usages. So band was originally running at EMBL, so based on EMBL stack from early 2022 until June 2024. It has been used for more than 20 courses during this time. Approximately, we have actually more than 700 users, and 90% of these users are from academia, and 95% is from within EU, UK, and Switzerland. So after June 2024, because EMBL decommissioned the OpenStack, so we moved the band to other places. So one in Tübingen, one in Cessna, with fewer resources. So far we have about 15 courses that use band as the main computational platform, and we have total number of 300 users. So because EMBL decommissioned OpenStack, so we can't have band at EMBL anymore, but we still need this virtual desktop experience. So what we do, we create another one because EMBL use Kubernetes to replace the OpenStack. So that's why we created the second type of virtual desktop called Bard. So Bard is cloud native, everything is Kubernetes based, and we maintain currently 2 instances. So the internal instance is for EMBL staff doing everyday image analysis tasks. And there's an external instance, and the hardware is also provided by de.NBI. So the external instance is also mainly used for external user and courses. So this is an overview of the Bard architecture. So different from band. So everything here is running on Kubernetes. And the difference, the main difference is in the middle. So there are 2 different configurations for Bard. One that you can configure the apps to run as a firmware container, which means you get a desktop pod. Everything application running in your pod use the same resources as what you have in your desktop. That's also possible to configure into another mode. So app runs as pod, so the same as desktop. In this case, then it's possible that your desktop running on node 1, but you have another application running on a different node. So it's sort of a waste of resource. So that's why we choose the first mode. And we have internal persistent volume to store the group shares and mount it to the internal Bard only. So this is the landing page of the Bard desktop. The flavors here, so you can only select, but you cannot arbitrarily set how many RAM CPU you want. So this is preset by the admins. This is one of the difference between Bard and band. And this is how the desktop looks like. So this is elastic in the desktop. So this is also what you will see in your browser. For data access in Bard is slightly a little bit different because it uses persistent volumes for internal Bard. So there are FS storage at EMBL. So our IT team just help us to mount this into the Bard. So the EMBL internal staff can use the same group share and scratch space. For external users, so upload data is via drag and drop. There's a size limit per file. For download, you require the separate applications. And we also provide desktop clients for accessing cloud storage. So, for example, we provide OnCloud and Omero client. and they are also containerized. Okay, this is a table compares the band and bar. So when to use band and when to use bar. So so we already mentioned that they based on different platforms, and then the configuration, one is user configured, one is predefined admins. So the access, so band is publicly available. So if the authentication is via either Google EGR checking or Life Science AI, so as long as you have one of these accounts, you can just freely use it without letting us know. But for bar, so it's not automatic now. So if you want to use bar, you have to contact us first, and then we issue you an account. Then you start using it. Oh, sorry. The container technology used to say in band is appended and then bar is Docker. So, and then for data access, we already talked about this. Okay, so now we cover this batch analysis and interactive analysis and a lot of different software solutions that we select to use at EMBL for these two different tasks. How do you choose which platform or which solution? How did we choose? How we decide that we use Nextflow, not Galaxy for the benzene, for example? So we think first of all, you have to think about this at the infrastructure level. So at the beginning, so this you have to decide whether your laptop is enough for analysis. So in most cases, it's not enough. Then you come to cloud. So, and then if you want to use cloud, the first question you're going to ask whether you want to deploy your own service or you want to use the existing services. Of course, if you deploy your own services, and then do you want to manage it? If you want to manage everything, like you want to manage the storage server, compute nodes, then we would recommend OpenStack. If not, maybe Kubernetes is better. So then, if you want to deploy or you don't want to deploy a service, then you have to go to search some public cloud which already has the service you want. And the next question is if you want to use public services and whether you want to put your sensitive data in the cloud. If not, then you need to search for a private solution. If yes, then again search the existing public solutions. So the next one, you have to think from the user point of view. So if you're a user, so how do you choose which software I will use for my analysis? So the first question we think would be useful is to ask whether you want to write a code or not. So if you want to write code, you're very comfortable at the scripting, then we will recommend using notebook environment, for example, Jupyter Lab, Jupyter Notebook. So if you don't want to use Jupyter and you don't want to write code at all, and then this is actually asking, do you want to build a batch analysis workflow or do you want to use interactive GUI? So if you want to build a batch, you maybe need to consider either Nextflow or Galaxy or maybe something else. Or if you purely want to use GUI, then we would recommend virtual desktop. So there's one more thing I mentioned is in this program is Open On Demand, so which is also a web browser-based system between user and the high-performance computing cluster. So Open On Demand can serve virtual desktop. So basically, you can get a band desktop via Open On Demand. And you can also run the batch analysis using Open On Demand. That's why we put here in both places. Okay, so let's recall this roadmap again. So basically, what we have covered today is the middle part. So the image format, storage, and this middle part of the tools that we use for analysis. So the one at the bottom right, we haven't talked about this, means the web applications. So that we have one application, Image Data Explorer. This is purely web-based. And so, yeah, lastly, so if as I'm part of this FDI, so let's say, for example, you want to start your image informatics roadmap for FDI or maybe for our own institutions, we probably need to think about the middle part. Yeah, lastly, I would like to acknowledge all of these organizations and teams for helping us setting up the environment and the infrastructures. Yeah, and that's end of my talk, and I'm happy to take questions. Thank you.
Thank you again to Jason for joining us today. And thank you very much to my colleagues, Daniel and Julia, the organizers of the webinar together with me. And of course, thank you very much to our audiences. And yeah, I wish you a nice day, and we see us again at the next de.NBI ELIXIR Germany and JJ webinar, I guess.
Thank you.
Thank you very much. Have a nice day.
Bye-bye.
Bye-bye.